Okay, I just started the recording. Okay, so like I said, we're going to go over, Shazia, did you have a question?
Yeah, I have a question. So I'm taking your Biology 1406 lecture, and I
Uh-huh,
have a teacher for lab, but I'm not
okay,
sure how to get to her. And I clicked on your lecture link that you have in your syllabus for this semester, and it
right.
brought me here, but I'm not sure if this is where I'm supposed to be.
Which teacher do you have?
Her name is Parisa Yazdi Rani. She has a really
Okay,
nice name.
so she is having virtual labs on August 24th. Let me see. Um, Hold on one second. I have to look too because I'm not 100% sure when hers is either.
Yeah, I tried to look at her schedule, but she didn't have a link or anything. I know she has Q&A sessions on Wednesdays, but I'm not sure if that's the actual lab.
She may just not have an actual lab time. It may just be that she expects you to work through all of the material and then come to the question and answer and just get your questions answered. Not, you don't have to do a live session, so it's not something that teachers are required to do.
Okay, yeah, so I
So
was confused about that.
yeah, I mean, it's pretty much the same material, so you're welcome to sit through this. Just know that anything I say that's specific for my lab might be a little bit different for hers, but in general, it's the same stuff. So it's up to you whether you want to sit through this or not.
Okay, yeah.
Okay.
I might just leave now, though. I'm going to email my other
Okay.
teacher.
Okay. Sounds good.
Thank you for your
I'll
help.
see you tomorrow though at 12:30. Okay, for lecture.
Yes, I'll see you.
Okay. All right.
Bye.
Bye. Anybody else have any questions before we get started? Okay. So what we're going to start with is just how the lab class part of this will work, and then tomorrow we'll talk a little bit about how lecture will work. Okay. So the goal for this particular, for our majors biology lab, is really to get you familiar with how to do research. It used to be in the olden days, like when I took biology, that you would come into a biology lab and they would present you with some sort of like canned, pre-programmed protocol. And they would say, it's almost like a recipe, right? They'd say, mix these things together and then look at the results, and then mix these things together and look at the results. And it was very much prescribed, and the instructor always knew what it was supposed to look like. And if it didn't look like that, then the student had to go back and start again. But what we were hearing from people going, advisors and professors in graduate programs and in biotech industries and in research labs, was that students weren't coming to them with the ability to think about designing their own experiments and getting asking the questions. They were learning how to follow a protocol, but they weren't learning how to think about it. And so about 10 years ago, there was this big push to allow undergraduate biology students actually do their own research. And so we've changed our class to give you that opportunity. So the first 8 weeks of the semester, we will present you with information in terms of techniques or some background information so that you learn some things. And then about 8 weeks in, we say, okay, you're going to get with a group, and based on everything that we've kind of learned so far, you and your group are going to design your own research experiment. You're going to come up with the protocol. You're going to come up with how to collect data. Then you're going to take that data and analyze it. And then you're going to do what a real scientist would do, and you're going to present that data in the form of a poster or a paper. And so that's how this class is going to work. Now, it's a little different this semester because we're online, but we're still holding to that same student-led inquiry-based lab, as opposed to just a canned lab that you just do what someone else already thought through and there's no real thinking involved. So that's our semester-long goal, okay, to get you familiar with research techniques and then give you that opportunity to design your own experiment. Our goal for today is to go over the syllabus and how the course is structured, and then we're going to talk about eukaryotic cells and some microscopy. All right. And that will get you set up for completing the rest of the activities on your own throughout the week. So let's start with the syllabus. So let me pull up the syllabus here so you can see it. I just realized my mouse is right here. Okay, here's the syllabus. Can you see that? So this is available to you on Blackboard, so you can pull this up and look. And you can see that it's numbered by week. So week 1, that starts today. And this is just the first day of the week. So for us, it's nice because our week 1 always is the day that our lab meets on Mondays. All right. And then it tells you the topic that we're going to cover. So today we're going to cover microscopy in cells, a little introduction. I'm going to let the lab safety part go to the online part. The next column here tells you what to do. So most of the weeks, there's just one thing to do. It's called a post-lab analysis. Typically, it's in Blackboard. It's like a quiz you would take in Blackboard, and it's proctored. So you have to have a computer that has a camera. So is there anyone here that still doesn't have a computer with a camera? Okay, good. If you still think, oh, I don't have a camera, I'm not going to be able to take these tests, it's a requirement that you have to have that camera. So we have, TCC has laptops with cameras in them that they will loan out to you for the semester. So if you're someone who thinks, oh, I don't have, continual access to a camera or something like that, you send me an email and I will get you in touch with the TCC loaner program for getting a laptop. Every once in a while, in addition to that post-lab analysis, there's some other little assignment, but very rarely. You can see here that assignment that week has an extra one. This week has an extra one. This one has an extra one. So it varies week to week. The post-lab analysis and assignments or anything else that's due are all due Sunday by midnight. So today's Monday. We're going to always review what you need to do, and then you have all week till Sunday when you get it completed. Okay, and you guys have me for lecture. That's the same for lecture. All my lecture stuff is due by Sunday at midnight. Okay, so I'm going to go ahead and get rid of this for a second. And I'm going to go here. So what's graded in this class? Because that's what typically students really care about. So there's a lesson outline for every lab. So let me take you there so you can see what that looks like. Oops, I'm in the wrong class. Well, I guess it doesn't really matter. Okay, so for when you go into the class, you'll click on our section, which is either 2004 or 2005, and you'll scroll down to laboratory materials. And then you'll see that each week, just like it was in the syllabus, is listed here. And when you click on the week, it's going to tell you what all you have to do. And then you have to keep scrolling down. So for example, here's a lesson on lab safety. There's an assignment on lab safety. Just want to make sure you guys are familiar with it. And then there's a week 1 lesson. So when you click on that week 1 lesson, it won't make you do that. That was just what it makes me do because I'm in teacher mode. You'll see that there is a whole lesson here. And embedded within this lesson are questions. So if some of you already did this, you'll see that there are questions in here that you have to answer. So you can see at the very top right here where it says score 0 of 11, you can kind of know how many you've done based on that score, okay? And at the end, there's going to be a submit button, then you have to hit submit. If you don't submit it, when you come back again, there's going to be a resume button that will allow you to submit. To pick up where you left off. All right, so for every lesson, for every lab, every week, there is a lesson like this, okay? The questions, those 11 questions, and some lessons have more, some lessons have less. The embedded lesson questions are worth 7% of your lab grade. All right, now just to kind of tell you, I have the same kinds of lessons in my lecture, and I have embedded questions in there, and they are worth part of your grade as well for lecture. Then you have those post-lab analyses and assignments. Those are worth 38% of your grade. This is not something that you want to blow off. Let me make sure I said that right, because that doesn't look right. Is that really 38%? Hold on 1 second. I feel like I said that wrong. Nope, I said that right. But the paper one part is wrong. Okay, give me 1 second. I just realized that this is incorrect. So it's supposed to be, the paper is 40%, doesn't add up. See, I knew it wasn't adding up to 100%. Okay, so the embedded lesson questions are 7%. The post-lab analyses and assignments are 38%, all right? Now those are timed and they're proctored, so make sure you study before you go and you take those, okay? Because you don't wanna mess up, you don't wanna rush and take it really quick and then not get a good grade when all you needed to do is just review your notes while you're taking it. We also will have 2 group presentations throughout the semester. One will be when your group gets together and you will propose what you're going to do for your research experiment. So it's kind of midway through the semester. And then there's one at the very end where you will present what your group found in your research experiment. Okay? And that's a group thing. So you'll work together a group and it's worth 10% of your grade. And then each of you will also write a research paper. So that's not a group activity. Everybody writes their own research paper. And notice here, that's worth 40% of your lab grade. Again, not something you want to blow off, okay? Now, you do get 3 chances to get it right. So you turn a draft in, I correct it, I give you feedback, I give it back to you. You make those changes, add some stuff to it. You give it back to me, I give you feedback, I correct it. I say I need more here, I need less here, whatever. I give it back to you. You still have another chance before you turn it in that last time for the points. So, you know, you really should be not, if you take it seriously, you shouldn't have to worry about research paper grade when you have 3 chances to get it right. Most people that take it seriously get an A or a B for sure on that research paper. And then at the end of the semester, your group will do, we're calling it a poster because in a face-to-face lab, it was a poster. For you guys, it's going to be a PowerPoint that you'll put together. When you're presenting your group presentation, you'll present it in the form of a PowerPoint. And so you actually get a grade on the actual PowerPoint itself. So you get a grade on the presentation and on the PowerPoint. So before I go to the next slide, you guys have any questions on the syllabus or what's graded? So every week you have to do the embedded lesson questions and the post-lab analysis. Some weeks there's an assignment, and then there are basically 2 weeks that there are group presentations. You have your paper due on certain days, and then the poster is due. So I can show you when the paper is due. Let me pull this up so you can see it again. So it says here, the week of October 12th, the first draft of your paper is due. And then we have a little bit of time between until the week of November 9th, you'll turn in another draft. The first draft is kind of, you don't have all the sections. This one has all the sections. And then I'll return it to you again. Let's see, this is not scrolling nicely. And then the last one is due during the week of November 30th, so right after Thanksgiving. Okay, any questions on where grades come from? Everybody good? Okay, so now we're going to move on to the actual lesson. Let me show you one more thing before we do. So in Blackboard, let's see where my Blackboard site went. In Blackboard, when you go into it, oops, I keep clicking on the wrong button. It disappears. Okay, when you go into Blackboard, make sure you look at that checklist for every week. So did you read the lesson and answer all the questions? I had the same kind of thing in my lecture. There's this checklist just to keep you on task. Did you complete the assignment called Lab Safety? Did you complete this lesson on eukaryotic cells? Did you take the practice quiz in Proctorio? Did you do the post lab on eukaryotic cells? So make sure you're doing all of those things. I tried to make it highlighted for things that were worth points. So questions in the lessons are worth points, assignments are worth points, post lab analyses are worth points. So if we go out, let's just look at one more week. So let's look at, we'll just scroll down to a later week in the semester. Let's look at this. No, let's look at this one. So this week, you can see the checklist is read and complete the questions in the lesson, do your post-lab analysis, and complete the first draft of your paper. And so if you scroll down from the checklist, you'll see lesson, post-lab analysis, and then the resources you need for your paper. And right here, this will be a Turnitin link. I haven't built it yet, where you'll turn your paper in. We always turn them in through Turnitin because that gives, it's a plagiarism check. Make sure you're writing your own paper. Okay, any questions on the syllabus or what's graded or kind of how it flows? Now, probably a question you have. I would say yes, that you can use your notes, but I would still study for the post-lab analyses, because if you don't study, it's a timed thing, and you can't just look up every answer. A lot of times it's like draw or graph or something like that. So it's not something that you have in your notes anyway, but you might have a little bit like, ooh, how do I label these axes on this graph? And so that might be in your notes. But yeah, you can use your notes. Okay, so one question that typically comes up on day one, and especially day one sitting here like remotely, how in the world are we going to do a research experiment? We don't have any experience with this, and you guys aren't even going to be in a real world. So how are you going to run a research experiment? Well, it turns out what we're going to do is you're going to get with your group, and you're going to come up with an idea. So I'm just going to kind of give you some ideas now, and later we'll talk about this again. Typically, I want you to think about something that may be good for the environment or good for the health of humans or something like that. So something that has a real cause behind it. So maybe you care about global warming. I saw that I had a couple people, I'm not sure whether it was a sex or not, that were they wanted to go into wildlife management because they wanted to help save endangered species, right? So maybe that's something that's really important to you. So maybe you want to address factors that contribute to global warming. So one thing, for example, could be like fertilizer runoff, right? So fertilizer runoff runs into lakes and rivers, and then you get these algae blooms, and the algae is great that it's growing overgrowing, but what it's doing is it's hurting the other organisms in that body of water. And so you're having the effect of global warming affecting the growth of the organisms in that water. So maybe you design an experiment around that. So maybe you say, all right, I'm going to take some photosynthetic cells like algae and some non-photosynthetic cells, and I'm going to treat them with and without fertilizer and see what happens, something like that, right? Maybe you're interested in the problem that's arising with antibiotic resistance. And so people take antibiotics when they don't really need to, and now we're seeing over and over that more people have resistance to antibiotics, which means they're becoming less and less useful in fighting bacterial infections. So maybe you're interested in like, could you, could essential oils serve the same purpose? Because people put essential oils in their food and things like that. So maybe you would take 5 different essential oils and then some bacteria and see how they grow in the presence or absence of the essential oil, maybe compared to bacteria grown with an antibiotic, so you can have a comparison. So obviously you can come together with your group, brainstorm some ideas, come up with an idea. You'll do some research online to see if other people have done similar things. And then yours will actually come up with a protocol. So here's what we propose to do. So we'll do it this way. We'll grow the cells, we'll treat them with the essential oil, we'll measure growth in this way, right? That's the part where it gets a little bit tricky, because normally you would come up with this protocol and then you would go in the lab and you would do the whole protocol, right? Well, we can't do that this time. So you'll come up with the protocol, we'll review it together, and then what we'll do is we'll provide you with data. So we'll say, here's some data from previous semesters where students had maybe done a similar project, and here's what they found. And then you'll use their data to analyze, graph, and then write your paper and your poster. Does that make sense? So don't worry about the fact that you can't actually come in. You can still do all of the brainstorming and pre-research together, because that can be done virtually. You can meet in virtually as a group. You can do research in the library online. So you can do those things virtually. What you just can't do is actually go into the lab and do the experiment. But we're going to provide you data as if you did, and you still have to come up with the protocol showing what you would have done. All right, and then you'll write your paper and your poster based on that. Oh, okay, so some people had a different link. Let's see. All right, do you know are you in that other link, Miles, somewhere? Okay, you got here, so can you copy the link that you used to get here, or do you need me to copy it? Okay, and then you'll tell them to go ahead and log in here. That's weird. I only have one. I never set anything up in any other class, but I guess sometimes things get weird. I don't know. Okay, so that's it for how the semester is going to work. If you have any questions or concerns, make sure you are communicating with me. I think when we're in this remote environment, one of the important things that we're all going to have to do better is communicate with each other. So email me, come to my office hours. I still have Teams, so you can communicate with me in Teams. You can get the information that you need. Don't ever feel like, ooh, I don't want to bug her. But that is my job. Like, I am here to be bugged, so go for it. Bug me away. Ask whatever. In my opinion, there are no such things as dumb questions, and all questions are good questions. So just ask whatever it is you have on your mind. Likely, a whole bunch of people are thinking the same thing and having the same question. So when I know that's something you are struggling with, I can help more than just you. Okay, so I'm going to go ahead and go to the next slide, and we're going to start the lesson for today. We will tell the people that are coming in. Oh, look at all these people coming in. Let me see if I can let them come in for a second. Oh, a whole bunch. Hi, everybody that just came in. Hopefully you can hear me. Sorry that you went to the wrong place. I will have to figure out how some people got one link and some people got a different link. I'm not really sure how that happened. Miles, do you think that's everybody? Okay, well, not yet. Okay, I see here there's a, it says, are there any online codes or anything required for the books that we purchase, or can you just rent a book? There are no online codes, no. You don't need to have any sort of access code for anything. So absolutely, you can rent the book. Let me just show you real quick. We don't have a book at all for lab. Everything's online in Blackboard. For lecture, this is the book that I use to build my lecture. This is the 3rd edition, but you can totally use the 2nd edition. Nancy, you can't hear anything. Hold on one second. And then this is the bigger version of that same book. And you can also buy this. You can use the 10th or 11th version of this. Either one is totally okay. The bigger version just kind of has more examples and some details that the smaller version doesn't have. I've taught out of both of those. If you really want to go free, you can use OpenStax, and on my syllabus I put the OpenStax chapters, and you can buy a hard copy of OpenStax too for about 40 bucks. So for me, I just want you to have access to a textbook, whether you rent it, whether you buy it cheap from an version, whether you buy it brand new and have one that's shrink wrapped and you have it brand new. I don't really necessarily care how you get the book, but I do feel like if you want to be a serious biology major, that reading the textbook is one of the first steps in getting really familiar with that material. Okay, well, I'm going to go ahead and get started. So those of you that came in late, we've been recording since the beginning, and all we really did was go over the syllabus. So after this, I'm going to post this recording, so just watch the first couple minutes of it to get what you missed. Okay, so we're going to move on to the lesson for today. So obviously this is biology, which is the study of life. If you did anything with our first lesson in lecture yet, you'll know that all living things are made up of cells, right? Cells are the basic unit of life. So some organisms are just one cell big, like a bacteria. Other organisms like us are trillions of cells big. Those cells all work together to form higher order functions. Cells were first described back in the 1600s, which is pretty crazy. That was a long, long time ago. Robert Hooke looked at a picture like this one down here at the bottom of the screen, cork from a tree, and he found that it looked like it was, when magnified, it looked like it was all these little compartments, and or like a cell, like a jail cell or a room cell, right? And so that's when he coined the term cell. And then people were looking at living things under magnification for many, many hundreds of years. And in the 1850s, the cell theory was established. So let me tell you real quick. Theory is, in science, we use the word theory when we have something that is supported by lots and lots of data and not really refuted. And so it's a way of saying this is, in a sense, fact. We don't really use that word in biology, but saying we have so much data supporting that this is true and no data suggesting that it's not true. So we're going to call it a theory. So we don't throw around the word theory in biology like we kind of do in our everyday lives. We kind of reserve that for something that is very much kind of broad in nature and supported by tons of data. So what does the cell theory state? It states one cell is the basic unit of life. It also states that all living things are made up of one or more cells. And then the last thing it states is that all cells come from pre-existing cells. So it doesn't address where the first cell came from. It just says once there was a cell, every cell came from the cell before it. And every living thing then was made of one or more cells. And if you get smaller than a cell, then you're not living anymore. Okay? So why do you think and use the chat, why do you think it's useful to study cells? Why do we start here? Why is this where we care?